So it was clear before, I'm not sure who's playing the role of Dan Aykroyd and Jane Curtin on this round. So Reza, we'll start with your comments to Norb.
I think what Dr. Kaminsky showed really clearly is that the mouse genomic responses and the human genomic responses are not the same. And the mouse and the rat and the mouse and the rat and the human are not the same. And to a certain extent, that shouldn't be surprising. What is perhaps more important with regards to this argument is the dose-response relationship for those differences. So while it's interesting to explore the details of a biological pathway and a particular response, if you're, for example, developing a drug for a pharmaceutical, when you're talking about the expansive set of chemicals that exist in the world, can these in vitro assays provide what we really need, or is the dose response even that one would imagine between the animals and the humans, does that fall in the same place, even if it's not predicting exactly the same thing? So to ask the question a different way, would a NOEL in a rat or mouse model for immunotoxicants be a effect dose for a human, applying the appropriate uncertainty factors on the risk assessment? What would we have more confidence in? The dose response and the confidence in a negative from an integrated animal model, or the dose response and the confidence in a negative for a human in vitro model? And wearing my hat, I would posit that the animal model is the right answer.
Norb?
There we go. So Dr. Rasselpour really does bring out a lot of very important points. I do agree that the in vitro models certainly have a difficult time recapitulating what happens in vivo. But the fact is that if we use the immune system as an example, this is one of the few systems where we can actually mimic the cell-cell interactions that occur in the intact animal. So we actually are able to recapitulate much of what you see in the in vivo situation in vitro. In terms of memory responses, which you brought up, the reality is that we cannot very easily measure memory responses in our rodent models or most of the animal models, because the animals have been housed in a situation that they're virtually not exposed to pathogens, so they have no memory cells to generate a memory response. Now that gets to what antigen we might want to use. Again, we have to select which one if we're going to do in vivo or in vitro. So I think many of the caveats that have been raised comparing in vivo versus in vitro, I think they exist in both situations. I think the other question is, at the end of the day, what are we really trying to measure? Are we looking for biological activity, which in some ways is what you're getting at, and maybe that may be more important than the apical endpoint that we're looking at. So I guess I'll stop there.
Okay. We have a question from the audience. Please identify yourself.
Donna Mendrick, NCTR FDA. So obviously, not only are mice bad to look at safety and inflammation, they're actually very bad at efficacy models of inflammation as well. There are a lot of horror stories of drugs that appeared to be efficacious in mice you get in the clinic and they fail. But given that, how would you go about replicating in vitro all the myriad interactions the immune system has in an intact being? Because that is going to be a very big challenge here. Skin sensitivity is just one aspect.
So I might have to take my hat off already. And what I would argue for is really a two-phased approach. Obviously, the animals still represent a very important model for safety testing. But I do think it is extremely important to begin to integrate also human-based models so that we can actually recapitulate or confirm some of the results we're seeing. One of the issues is, you could say, well, animal testing has done us very well, especially for the immune system, because we really can't point to many examples where chemicals were tested and came up negative and then were used, and do we see problems? My argument would be, how do we really know? Because suppression of the immune response may manifest itself in that, maybe you had the flu this year where you wouldn't have had it. So we don't really know. So you're right, to recapitulate the entire immune system in a single assay cannot be done. Many of the assays that we conduct in vivo can be done in vitro and can be done in human cells. Now, the challenge there is using primary cells, and in fact, you do see differences, at least in the control responses from donor to donor. But those assays all can be done in vitro, or most of them, just not in a single assay.
Stan Barone, I'm going to sort of throw something in that's a little bit orthogonal, and that is you talked about the transcriptomics comparison between species. I think there's a lot of problems with just transcriptomics versus function, and so I was wondering if you could comment on comparison of proteome and protein function between species.
I think it's to me again. So, could you restate that just one more time?
Well, here, let me give you the context. Previously, I actually did some research on this subject, and we showed that developmentally, if you looked at different parts of the brain at time course, and you looked at the transcriptomics and you looked at the proteome, you could not predict from the transcriptomics the cytokine expression and the neurotrophin expression of a whole family of proteins based upon that time course of the transcriptome and the proteome, and in the same species. So you're trying to go across species, and I'm wondering, again, about this caution about comparing transcriptomics when you really have possibly different mechanisms between organ systems for the different cytokines or the different genes.
So there's no doubt that the transcriptomic signature is going to give you a definitive signature of what proteins are made, absolutely. But on the other hand, we do know that those genes do control protein expression. And so if we see that they're regulated differently across different species, then obviously the experiments need to be done, but that would also then lend you to believe that also the regulation of those genes, which ultimately leads to the protein, is going to be different too. Now, the bigger question, and I don't think this has been investigated extensively, are the signaling pathways that ultimately lead to the suite of transcription factors that regulate these genes, and has throughout evolution, has that also changed? And so, we know that they're ubiquitous signaling pathways, and there's, I forget the actual number of these, but you can, depending on how you slice and dice this, you probably get it down to about 15 different kind of general signaling pathways. The question is, are they activated in the same way? Are they regulated in the same way across species? Because clearly, the gene regulation piece is also very dependent on those signaling pathways upstream, and how much of this is conserved also across species is very important to know.
Norb, I've got a question about your conclusion that that cross-species gene expression profile was not concordant, because you said that group of 28-ish genes that was concordant was AHR-mediated gene expression, which seems to be exactly what you would expect to be conserved across species, and that would seem to be a very specific predictor set then.
So I would agree and disagree. So I would agree that yes, that those 28 genes are clearly AH receptor regulated, and so they clearly are part of that pool across the species. But if you can only pull out 28 that actually go across 3 species out of what was it, over 2,000 in the mouse and over 700 in rat and 500 in human, if you only did one species, you would have no idea what they are. And in fact, even out of the 28, we're still trying to find the 1 or 2 out of that that actually have anything to do with regulation or adverse regulation of the B cell. So in fact, even though we've got it down to 28 genes, we still might not have the right gene or genes that are actually altering the ability of the B cell to become an antibody secreting cell. So, and now as we dig in more, it looks like the mechanism between mouse and human is very different when we look at the apical response at the end. So maybe all of this is an exercise in futility, but a lot of work.